Biotechnology and its applications. So biotechnology applications in agriculture. Next one, biotechnological application in medicine. Third one, transgenic animals, and fourth one, ethical issues. So first one, biotechnological applications in agriculture. There are 3 points. Then genetically modified organism, GMOs. So what are these? So plants, bacteria, fungi, animals whose genes have been altered, whose genes have been altered whose genes have been altered by manipulation are called genetically modified organisms, that is GMO. Okay, now under that, so in the biotechnological applications in agriculture, we study the BT cotton. So what it has an insecticidal protein. What are these? Now these are the crystals contain a toxic insecticidal protein. Okay, there is cry. So what is that? The toxin is coded by a gene named cry. So cry it is a gene which codes for a toxin. Now there are cotton bull. Now cotton ball destroyed by bullworms, a fully mature cotton ball in the second diagram. In the first diagram it shows destroyed by bullworms. Now pest resistant plants. There is a process that is RNA interference that interferes the mRNA, the silence the mRNA. This is RNA interference. Pest resistant plants, the process is RNA interference. See in the diagram. So host plant generated double stranded RNA triggers protection against nematode infestation. Okay, so host plant generated double stranded RNA triggers protection against nematode infestation. Now in first A, roots of a typical control plants. So roots of a typical control plants. Now in the B, transgenic plant roots 5 days after deliberate infection of nematode but protected through novel mechanism. So in the second diagram, we are going through the novel mechanism, that is RNA interference. So this is it. Now biotechnological applications in medicine. Now genetically engineered insulin. We study the genetically engineered insulin. So how is that? There is C peptide. What is that? It is an extra stretch. Now maturation of proinsulin into insulin after removal of the C peptide, that is extra stretch to be simplified. Now in the first, it is proinsulin. Now proinsulin contains A peptide, B peptide, and A peptide, B peptide, and extra stage, that is C peptide. Now in the mature one, that is insulin. So from proinsulin it is maturing into the insulin. So there is removal of C peptide. So you see pre-C peptide, or A and B peptide are bonded by disulfide bonds. So this is it. So this is the insulin. This is the structure of insulin. Now gene therapy. There is molecular diagnosis. Transgenic animals. What are these transgenic animals? The animals that have had their DNA manipulated to possess and express an extra foreign gene are known as transgenic animals. Okay, now we study them for normal physiology and development, study for diseases, biological products, vaccine safety, chemical safety testing. Now ethical issues. There is organization, that is GEAC, Genetic Engineering Approval Committee. Now it is appraisal committee. Okay, the Indian government has set up organizations such as GEAC, Genetic Engineering Appraisal Committee, which will make decisions regarding the validity of genetic modified research and the safety of introducing GM organisms for public services. Now this is it. Now there is another term, that is biopiracy. So it is a term used to refer to the use of bioresources by multinational companies. It's kind of robbery and other organizations without proper authorization from the countries and people concerned without compensatory payment. They don't give you anything, they just take everything from you. That's the biopiracy. So biopiracy is the term used to refer to the use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment. This is it.